Hello guys, welcome Kwambonyash. This is DMLT2 2022. Welcome. Today we are going to learn about hematological stains. These are vital stains in hematology lab. We are going to look into the principles, theories, composition, and type of stains used in hematology lab. We are going to start with they are called Romanistic stains. We use them in day to day in our labs. These stains are used in hematology and cytological studies to differentiate cells in microscopic examination of blood, bone marrow samples. These stains are also applied to detect presence of parasites in the blood, such as malaria parasites. There are many various Romanistic stains, types that apply the same principle. The principle of Romanistic stains. These stains are neutral. They are made up of oxide methylene blue, azole dyes, and eosin white. The azole are basic dyes that bind to the acid nuclei, forming a blue-purple color. The acid dye eosin binds to the alkaline cytoplasm, forming a red coloration. Let's now see the types of Romanistic stains. The types of Romanistic stains. The first stain is Leishman stain. This stain was discovered by William Leishman using polychrome methylene blue and eosin and methanol solvent. The stain is used to differentiate and identify white blood cells, malaria parasite, and trypanosomes. The use of methanol acts as a fixative, preventing perforation. The preparation of Leishman stain. The methylene blue is polychromed by heating 1% solution with 0.5% sodium carbonate at 65 degrees Celsius for 12 hours, after which a further ripening is allowed to proceed for 10 days before it is mixed with an equal volume of 0.1% eosin blue. Number 2, we have the Jamesha stain. It is also used as a differential stain for various blood cells. The erythrocytes, these are the red blood cells, the platelets, the leukocytes, and cellular components such as nucleus, and the cytoplasm. It is made up of acidic and basic dyes of eosin Y and methylene blue, hence it is an azole stain. It is an azole stain. This produces blue-purple color staining of the nuclear and the red-pink coloration of the cytoplasm and the cytoplasmic granules. Let's now see the composition of Giemsa stain. Giemsa stain can be prepared in-house using Giemsa stain powder or can be commercially obtained. The basic ingredients for both are the same. However, dilution can be made depending on the use or the manufacturer's instructions. The composition. It has ingredients and the concentration of the ingredients in grams per liter. We have the Giemsa powder, which is 7.6 grams per liter. Glycerol 500 ml. We have methanol 500 ml. Let's now see the preparation of Giemsa stain. Weigh the required amount of powder stain. Transfer to a clean, dry 1 liter capacity of bottle. Add methanol and mix. So the first component is the powder, then the methanol, followed by mixing. Measure and add glycerin to mix. So the next is to add glycerin and to mix. So you add glycerin and then you mix well. Place a bottle of stain in water bath for 50 to 60 degrees Celsius or at 37 degrees for up to 2 hours with frequent mixing. If there's no provision of water bath, you can leave it in 37 degrees for 2 hours with frequent mixing. So there is this frequent mixing which should be noted. Label the bottle and store it cool in a dark place with a firm stopper. The next one is rats and rat Giemsa stain. It's another Romanistic stain which was discovered by James Homer rat by modifying the Romanistic stain. So it is a modification of a Romanistic stain. Rat stain used heated methylene blue, which produces polychrome methylene blue combined with eosin Y. The heated methylene blue with eosin Y is allowed to precipitate to form an eosinate, which is then dissolved with methanol. This defines the procedure of the red stain. When Giemsa stain is added to the rat stain, the color brightens to a reddish purple in the cytoplasmic granules. These are hematological stains that are used to differentiate blood cells from peripheral blood film and bone marrow aspirate and even urine samples. Majorly, they are used to differentiate stains. Majorly, they are used as differential stains for the various types of white blood cells. They can be used quantitatively for white blood cell count in persons with parasitic blood infections such as malaria and disorders like anemia. So what have we learned today? We have learned the various types of blood cells, blood hematological stains, and we have seen they are called Romanowski stains. There are many types of Romanowski stains. We have the Gemsha stain, the rat, and the rat Gemsha stain. We have the May granule stain, and then we have the Leishman stain. So they share the principle. They also share the components. So their main aim is the ability to stain the nucleus and the cytoplasm. So thank you for coming. Let us meet in the next lesson. Bye bye.